Okay, so this is just a short video introducing the next topic. The first part of that is looking at energy transfers, so linking in from our work with photosynthesis and where biomass comes from, and how we can talk about that in terms of productivity. Okay, so first of all, a little recap of photosynthesis, just to get us thinking about where biomass comes from. So as you can see, just a little recap of the Calvin cycle here, where we've got our ribulose bisphosphate fixing the carbon dioxide onto GP, which converts to TP, mostly regenerated, but of course, after every 6 turns, we do actually get a glucose molecule being formed. Now, what actually happens to that in the plant? Well, of course, we know that it's stored as starch, storage polysaccharide, or it's also converted into cellulose for the use in cell walls. It's also, though, via various biochemical pathways, converted into other useful organic biological molecules such as the amino acids, the lipids, and the nucleotides, and obviously goes off from there to form the larger macromolecules. Of course, some of the glucose made is just used directly by the plant in respiration. And it's the other fates, really, the starch, the cellulose, stored as lipids in seeds, nucleotides, amino acids for proteins, nucleotides for DNA. Those biological molecules are actually the basis for biomass in a plant. Okay, because of course the glucose, if it's used in respiration, it's not going to be used in making that plant grow or get any heavier. But all of the other ones are going to become part of the plant, and it's those things that we're talking about when we talk about the word biomass. Now, unfortunately, to understand this topic, obviously there's quite a few key words that we need to make sure we are familiar with. Quite a few of them I am sure you'll be more than happy with from your work at both Key Stage 3 and Key Stage 4. Okay, so first of all, obviously we've got our producers, should be more than happy with that one. So that's all the organisms which are able to photosynthesize, i.e. they can make organic substances, which we know from the previous slide is what the biomass is, and they do that using the light energy from the sun, water and carbon dioxide. Of course, they also need mineral ions. In the case of chlorophyll, they use magnesium in there, and be aware there are some types of organism which can actually use hydrogen sulfide in photolysis. So that's obviously instead of water, so splitting water, they're able to actually split hydrogen sulfide, and therefore their product is sulfur as opposed to oxygen gas. So yeah, that's a specific type of prokaryote. Just mentioning this, it's not in the textbook, but I've seen that as an exam question before. Application, of course. Then, of course, we have the consumers. Again, a word you should be familiar with. They're the organisms which cannot get their energy directly from using sunlight, so they have to obtain that from feeding on other organisms. And again, you'll be familiar with the terms primary, secondary and tertiary. So the primary consumers are always towards the bottom of the food chain, those which feed directly on plants, your herbivores, and your secondary and tertiary, there actually are sometimes quaternary as well. Consumers are feeding on other organisms. Generally, those are like predators, animals which are predators, but you can also get scavengers, parasites, those types of things which just feed on other organisms. Okay, so the third and final group of organisms are the saprobionts. Okay, so this is probably one, I've highlighted it in red just because there might be quite a lot of you that have never heard that term before, and I will be going on in a later video to talk in a bit more detail about this category of organisms. So they take part in something called saprophytic digestion. But in a nutshell, it's the decomposers. Okay, so we're talking predominantly about the fungi and the bacteria. Okay, so their role is just to basically recycle organic compounds when an animal dies or a plant dies. So they're breaking down the organic material found in dead organisms, of course bacteria would be included in that, releasing the goodies, basically the minerals, other elements which can be absorbed by plants and obviously therefore recycled by them. And again, we will go on specifically in this unit to look at the phosphorus cycle and the nitrogen cycle. Okay, and finally we need to just make sure we're all clear, which I'm sure you are, probably this at Key Stage 2, that obviously you understand what a food chain is and how that differs from a food web. So arrows represent the flow of energy between each stage or each organism, and we call each level at which that organism exists as a trophic level. So the producers are one trophic level, the primary consumers are the next trophic level, and so on. But as I'm sure you are all aware, in practice, it never really, you very rarely get a single food chain. You generally find that they exist as food webs. Where you've got lots of food chains all interacting with each other. So generally one thing will eat more than one type of organism, and so on. Okay, so as always, it's good to just be aware of how they might actually talk to us about the use of biomass in exam questions. It tends to generally be in the form of understanding what dry biomass is. Some of you might remember way back at the start of year 12, we actually did an exam question right in the first unit of biological molecules, looking at the dry mass of cellulose. And because of the amount of water in a plant is variable, it's no good scientists, taking organisms and measuring their mass and then using that to infer biomass. So actually it's important, what they do is they dry it in an oven and they keep drying it. They check its mass, probably once every 24 hours or so, and to ensure it's at a constant mass. And once it's there, they know that all the water must have been evaporated. Also at that time, it's important not to have the temperature too high, because that will ensure that you're not actually causing any chemical reactions to happen. You're literally evaporating away the water. But it's also important to note the big downside of this, and that's that obviously if you're going to dry something in an oven, then clearly the organism won't be alive at the end of it. So for that reason, we generally only do it on quite small numbers, and therefore there's the risk that it's not actually going to be representative. Okay, so the units are generally, well, it depends on the area being sampled. If it's, your sort of standard layer quadrat down on an area to sample, such as, moorland or rocky shore, what have you, the units will be in grams per metre squared. Okay, and obviously those grams will be the dry mass. If, however, you're sampling in an ocean, then the units would need to have a volume, so it'd be grams per metre cubed. Clearly you want to be looking at the entire area of whatever it is that you're sampling. And of course it would really be very important to make sure that you've also taken into consideration time. Okay, so scientists use a technique called calorimetry to try and estimate or work out the amount of chemical energy stored in this biomass that we've just worked out. Okay, and you can see here on the right, obviously, is a picture of a sort of modern day bomb calorimeter, which is the name of the piece of equipment that they use. And there on the left, a little bit more of a complicated version. I've got a more simple one, which I'm actually going to talk you through in a second. But in a nutshell, that's what it looks like inside. Basically, they just heat a sample of this in pure oxygen, have a water bath that is then heated up as the energy is given off by the burning thing, and then by measuring the change in temperature, they can work it out, because obviously that we know the specific heat capacity of water, we can work it out. So I'm just going to explain that to you using a more simplified diagram. Okay, so here's a more simple version of what a bomb calorimeter looks like inside, and as mentioned, here's our sample, our dry biomass, as previously explained, and it's actually heated up using these wires, these ignition wires. So that's just a little bit like a light bulb, and inside here we've got some pure oxygen. Okay, so as the sample combusts or burns in oxygen, clearly it's going to be giving off a lot of heat energy, and that heat energy is used, or will be converted and used into heating up the water in here. Clearly you can also see we've got a thermometer in there, and of course the thermometer can be used to measure the start and end temperatures, and we can use that in the specific heat capacity equation. If we rearrange that, because we know how much heat energy is required to raise the temperature of 1 gram of water by 1 degree C, as long as we know the volume of water and we know the temperature change, we can calculate the energy released from the mass of burnt biomass in units such as kilojoules, so the energy, per kilogram of biomass. Sorry. Obviously it's really important that there's insulation around the edge, and I guess that's the sort of thing they could maybe talk to you about in an exam question. Why is it important that we have this insulating layer? Clearly we want to make sure all of the energy released from this sample is used to heat up the water, and we want to measure that change. We don't want to be losing any of the heat to the environment. Okay, so I'm actually going to go on now to explain to you what we mean by the term productivity, and actually just for ease of uploading and file size, I'm actually going to have that on a second video. Okay, so please go on now to click on the second link and ensure that you understand what we mean by productivity.